Good morning, everyone. Welcome again to our wonderful annual occasion, our breakfast. I'm Janet Rossand. I'm President and Scientific Director of the Gairdner Foundation, and I'm really excited again to be with you for what we call the big reveal. Although if you picked up your Globe and Mail on your doorstep this morning, or you picked up the leaflets, you already know the answers. However, this is the announcement of the 2019 recipients of the Canada Gairdner Awards, and another amazing group of scientists that we're recognizing. I'm particularly excited because this is the 60th anniversary of the Canada Gairdner Awards. Built on the initial vision of Jim Gairdner, who you saw on the video, these awards have really continued to grow in prestige over the years. During 60 years, we've awarded over 380 scientists and researchers from over 35 different countries for their incredible work. And I'm pleased to be announcing 7 more individuals who will join that group today. Got a great crowd with us. Thank you all for being here to celebrate this occasion. I'd also like to welcome those of you who are joining us on live webcast from around the world. Welcome. Thank you for tuning in, and we know that people do tune in. I encourage you all, please, to share the news on Twitter and Facebook using the hashtag @Gairdner and the handle @Gairdner Awards. So this year, we're going to be hearing from each of our awardees, either in person and or by video, about their groundbreaking work. And at Gairdner, we really pride ourselves on the caliber of the awardees. That's what the prestige is all about. They're selected through a very robust adjudication process, which involves leading scientists from around the world. So I'd like to thank the members of the Medical Review Panel, the Medical Advisory Board, the Global Health, and the Whiteman Award adjudication committees for all their hard work in selecting this year's outstanding cohort. It's an amazing, fun, but very difficult task. I'd also like to take a moment to thank our long-term supporters at Gairdner. The federal government, through CIHR, has been a long-time partner, and their commitment is really essential to the prestige of the awards, but also to our coast-to-coast programming that gives students, trainees, faculty members, and the public access to all this groundbreaking science that's changing our world, encouraging students to take up careers in STEM and engaging the public on how science is affecting them. The Gairdner Foundation is also strongly supported by our media partner, the Globe and Mail, who help us bring our laureate science to the public. So as I say, check out your paper and their website today to see a great feature on our laureates. Please also check the journals Cell and Lancet, because they also have articles on this year's laureates. So today, we're very fortunate to have Dr. Stephen Robbins here with us on behalf of CIHR. And Dr. Robbins is the Scientific Director of the CIHR Institute of Cancer Research and the current co-chair of the Canadian Cancer Research Alliance. He's a professor at the University of Calgary in the departments of oncology and biochemistry and molecular biology. So please join me in welcoming Dr. Robbins as he shares a few words about Gairdner's partnership with CIHR. Thank you.
Thank you very much, Janet, for those kind words, and good morning, everyone. It's really great to be here and a real honor. In fact, I'm a little in awe of the people that are in this audience, and Dr. Till sitting at my table as I did my graduate work looking at stem cell transplant, and it's amazing, all the recipients. And I think we're at about 387 awards that have been given out over the years since 1959. So it's my great pleasure and honor to be here on behalf of the Canadian Institutes of Health Research, or CIHR, and our new president, Dr. Mike Strong, and he sends his best wishes and congratulations to this year's recipients. So at CIHR, we've had a long-standing interest in supporting the best science in this country, and I think this event is an example of the kind of research that we see and the kind of impact that we can have as researchers in Canada, both here in Canada, but also our global outreach. As people might know, we are set up at CIHR as 13 virtual institutes that are speckled across the country, and that through this distributed institute system, we try to keep associated with all the researchers within this country. It's been a challenging time over the last number of years. I've been now 6 years as a scientific director at CIHR, and we're looking forward to the evolution of moving forward with strong support from our federal government. I think that the mandate and vision of the Gairdner Foundation is very similar to what we have at CIHR, and I think we align very nicely. Supporting scientific excellence and recognizing scientific excellence, having an impact on the health of Canadians and beyond, and supporting the next generation of researchers, something that I feel very passionate about in my role as a scientific director at CIHR. We also are very proud of and supportive of the mission and programming. And I was looking at the little pamphlet here, the little infographic, and really looking at the type of outreach that the Gairdner Foundation does. 7,500 students every year are supported, and 20 universities across Canada are involved. And we even, the Gairdner Foundation even supports at the level of the high schools. Over 100 different high schools are involved in this program. The public lecture series that brings the brightest minds in science to a public audience. I think that's another area of which Canadian researchers need to continue to strive for, is that the communication to our public, the importance of research that we have and how it can really change how we are as a Canadian society. The other aspect is convening leaders, and this would be a very great example of convening leaders within this room for breakfast. And then inspiring the next generation, as I said, which I think is a real essential aspect. Now, I was lucky to have trained in San Francisco with Mike Bishop in the early 1990s, and he was a recipient of one of the Gairdner Awards in 1984, along with his close colleague Harold Varmus. And he always said he was an honorary Canadian, and I think now he even wishes more to think of that. As Janet said, this is the 60th anniversary of the Gairdner Foundation. I think it was actually established as a foundation in 1957, but the first awards were in 1959, and as I said, over 387 awards have now been delivered, and I wish everyone the best. Congratulations to the foundation under the leadership, John Dirks, Janet Rosant, and everyone. And look forward to hearing the reveal, although I guess it's out in the Globe and Mail this morning. I was on Calgary time, and so that was a little early for me to take a look at the Globe and Mail. So thank you very much for the opportunity to say a few words on behalf of CIHR.
Thank you, Steve. That was great. And we really do appreciate our ongoing partnership with the federal government and CIHR. So let's begin this morning's award with the announcement of our Canada Gairdner Whiteman Award. And the Whiteman Award is reserved for a preeminent Canadian scientist and leader. This year's awardee was recently inducted into the Canada Medical Hall of Fame and is not only a leader in her field here in Canada, but around the globe. The 2019 Canada Gairdner Whiteman Award goes to Dr. Connie Eaves, who is with us here today. Her citation says, for her pioneering work and leadership in the study of hematopoietic, mammary, and cancer stem cells, and her dedicated advocacy for early career investigators and women in science. Connie Eaves is a distinguished scientist at the Terry Fox Laboratory at the BC Cancer Agency and a professor in the Department of Medical Genetics at the University of British Columbia. And Dr. Eaves' research really throughout her career, as she'll probably tell you about in a minute, has focused on the properties of normal stem cell hierarchies in the blood system and in the mammary gland, and how those pathways get hijacked in leukemia and breast cancer. But in addition to all the accolades she's received for her research, she's really recognized for her exceptional commitment to the training of more than 100 scientists from around the world, including many who themselves now hold senior leadership positions in science. She's a passionate advocate for the advancement of women in science, a commitment that led to her recognition as a Status of Women Canada pioneer. She continues today to apply cutting-edge technology and elegant experimental design to the most pertinent problems in stem cell biology and cancer research, consistently contributing to the ongoing pursuit of cures. So we're going to hear from her in the video, and then, because she's here with us, we'll hear from her in person. So Dr. Eaves' video.
What challenged us and what we thought was going to be the most interesting area to pursue was, what are the cells and what are the properties of the cells that give rise to blood cells? And what goes wrong in these cells or their immediate progeny that give us leukemia? We started out to address those questions by trying to develop quantitative methods that would be robust and reproducible over time so the whole world could use the same procedures and replicate any results. And that kind of approach gave us insights into some of the properties of those cells that are abnormal, and now we're trying to understand what the molecular basis of those changes are. I think what we will be recognized for a long time are the methods that we've developed that have made it possible for many investigators all over the world to address the same questions, because this is a big area of unknown still. And the fact that it could be extended not to other tissues like the mammary gland and becomes relevant to many areas of cancer. Almost all of our advances in treating patients have come from basic scientific understanding. And so that remains, in my view, a very, very important aspect. We are not on the verge of curing everything, or even hardly anything. We're on the verge of making big advances. But yes, we have initiated clinical trials. We've made discoveries about the presence of normal cells in patients with leukemia that have had a big impact on the field. So both areas are important. I never thought that I was in the league of being recognized for something of this magnitude. And I'm very grateful, because it is an opportunity to speak out and make other people feel good about wanting to pursue a career in research and science and promote it in Canada and the world.
Well, good morning, everybody. I don't know what to say. I just love Canada. Anyway, let me start by saying happy birthday to the Gairdner Foundation. I learned last night that the 60th anniversary, long gone for me, is a very special one. And so it's incredibly wonderful to be here to celebrate a birthday party for an organization so important. My hairdresser on Sunday said, "You got to say you're proud." So I'm very proud and incredibly honored to acknowledge this amazing recognition. And I speak really on behalf of a huge team of trainees and colleagues, not me, who have worked together over many years, inspired by an area of science that was born in Canada. I speak, of course, of the conceptual basis of stem cell biology. Stem cell biology has now revolutionized ways to treat patients with some forms of leukemia and immune diseases, and has spurred the development of a new age of regenerative medicine. However, this is just the end of the beginning. There are still many complex problems to solve. And I therefore very much hope that this award will signal to young women and men everywhere, I see all of you out there, maybe you want to come and do a postdoc, that further pursuits of their interest in science is what is needed most to build on past advances and make the world a better place. Now, I'm supposed to talk a little bit about me and what I do. So, in 1905, my paternal grandmother, at the age of 16, ran away on her own from the pogroms in the Ukraine to come to the United States. So that's quite a heritage. She wanted all her children to become academic superstars, and also passed that imperative on to me. Since my dad and older brother were destined to become brilliant mathematicians, I, of course, wanted to do something completely different. So I entered the realm of biomedical science, where I saw tissue development, cancer, and genetics, that's back in the 60s, as the most fascinating uncharted areas to pursue. And so I headed in that direction as fast as I could. I soon realized the power that could come from developing precise ways to analyze different types of primitive cells using quantitative clonal methods, and began to chart a course of investigation based on that idea. Alan, husband and life partner in science, kept us trying to apply our findings to leukemia as fast as we could make that leap. And 20 years later, I got the idea of doing the same thing for mammary cells and breast cancer. Who would have guessed in 1990 that a single cell, a single human cell, could produce all of the blood cells in a person for a year? Or regenerate a complete mammary gland that produces milk? Or that certain leukemic cells can go into a deep sleep that makes them insensitive to the very drugs that we use still today to reduce the leukemia to an undetectable level. These discoveries are examples of what a group like ours can find with no sense of boundaries and determined to address the biggest challenges out there. No doubt you will detect my grandmother's legacy at work. On a personal note, I've had an incredible life experience. I wake up every morning in love with my life, my family, my colleagues, and the principles by which we find out how life works. And I've had additional important lessons along the way. While studying cancer for many years as a scholar, I also worked closely with the Canadian Cancer Society, and I learned that progress is as much about listening as teaching. 13 years ago, I myself was diagnosed with breast cancer, and I'm a double mastectomy survivor. And I experienced the fear of being hit and with such news, and how to get over it. But I'm fine now. So you can come as a postdoc, not worry. And what is now exciting our research team? While the rest of the world is busy studying the complexities of cancer by analyzing patient samples, we have discovered easy ways of making it experimentally in human cells from normal people. So we can produce human leukemias and breast cancers quickly and reproducibly, starting from the very same normal cells in which these diseases arise in people like you and I. And the expectation is that these new models will make it easier to nail down the key events that cause these cancers to develop in the first place, and then learn what might be new therapeutic vulnerabilities. That's so I can get my grant funded, Steve. Wish us luck. We are in it for the long haul. And again, I am so incredibly grateful to the Gairdner Foundation and all of you who support it, and all of you who will support it. You engender the sort of encouragement that scientists everywhere need and treasure. Thank you so much.
Thank you, Connie. Another proud Canadian, and there's a room full of proud Canadians here this morning. So now it's time to move on to our 2019 Canada Gairdner International Award laureates, 5 international awardees. And these awards recognize seminal discoveries that have had a broad impact on biomedical science and health outcomes. And the first of the 5 2019 Canada Gairdner International Awards we're going to talk about today goes to Dr. Susan Band-Horwitz, who is also with us here today. The citation reads, "For defining novel mechanisms of action and resistance of drugs of natural product origin, most significantly Taxol, and promoting their use in the treatment of cancer." Dr. Horwitz is Distinguished Professor and Rose C. Falkenstein Chair in Cancer Research in the Department of Molecular Pharmacology at Albert Einstein College of Medicine, New York. And her body of work is really an inspiring story of determination. Although people were not really interested in Taxol when she began her studies, today, because of her understanding of its mechanism of action, it's an important anti-tumor drug for the treatment of ovarian, breast, and lung carcinomas. So here to tell us a bit more about Dr. Horwitz's work is Dr. Rama Koka, Senior Scientist at the Princess Margaret Cancer Center at the University Health Network, and Professor in the Department of Medical Biophysics and the Department of Laboratory Medicine and Pathobiology at the University of Toronto. So please welcome Dr. Koka.
Thank you, Janet. Good morning, everyone. It's lovely to be here today and share this news with you. I'm very excited to be here today to announce and introduce the first 2019 Canada Gairdner International Awardee and speak to you about her field of cancer research, molecular pharmacology, and the development of the drug Taxol. Dr. Susan Band-Horwitz set out to determine the mechanism of action of Taxol in the 1970s. Through her work, she discovered that Taxol binds to microtubules in cells, paralyzing them, stabilizing them, and leading to cell cycle arrest in the mitotic phase with the subsequent tumor cell death. This body of work has enabled the successful translation of Taxol into the clinic. So today, it is one of the most frequently prescribed drugs in the world for treatment of ovarian, breast, and lung cancer. Despite the fact, as Janet said, there wasn't much interest in Taxol when she first began her studies. In addition to being given to millions of cancer patients worldwide, Taxol is actually also used in the preparation of cardiac stents for cardiac disease, and also it has become a fundamental tool for scientists who are interested in the study of microtubule structure, dynamics, and function. So as a freshman at Bryn Mawr College, the young Susan Horwitz took a mandatory course in biology, and that course inspired her to take on science instead of pursuing history. Lucky for us. She went on to get a PhD in biochemistry, and as a young woman, she shouldered the responsibility of raising a family and yet followed her passion for scientific discovery, continually developing sophisticated Taxol analogs with her colleagues to unravel how precisely Taxol function. She also, in parallel, worked out the mechanisms that cells use to acquire resistance to this important drug. And beyond Taxol, she's actually worked on a number of other drugs and worked out their mechanism of action. I love the way she goes on to describe the structure of Taxol. She says, "A structure only a tree would make. Chemists may not think of this, but nature does." And I think that really captures the essence of her commitment to Taxol and advocacy of Taxol. So in summary, her work is an example of dedication, determination, and scientific excellence. I am thrilled to be here, and I'm thrilled that Canada Gairdner Awards Foundation recognizes her excellence, and also that I had the opportunity to introduce her today. And thank you again. And now before she comes to the stage, please take a moment to watch the video of Dr. Horwitz explaining her work in her own words. Thank you.
Well, I've always had a long-standing interest in natural products as a source of new drugs for the treatment of cancer. So when I was introduced to Taxol, no one had any idea of how this drug kills cells. So I set out to try to figure out what is the mechanism action of Taxol. It was a question because this was a drug that we knew inhibit the proliferation of cells growing in tissue culture, and we also knew that it inhibited the growth of B60 melanoma, a tumor in mice, but nobody had any idea of how this was happening. We really knew very little about this molecule, but what we found out was that this molecule binds specifically in the cell to part of the cytoskeleton of the cell. So there are many components in a cell that are part of the cytoskeleton, but we particularly were looking at microtubules. And when we began to look at these cells by electron microscopy, what we saw was that there were many bundles of microtubules in the cell. And this was extremely exciting because this was a new mechanism of action that had not previously been described. I like to say that was a hallelujah moment for me when I realized that this small molecule, Taxol, had a new mechanism of action for an anti-tumor drug. And of course, since the drug has been approved, it's been given to millions of patients throughout the world. So I think it has had a definite effect on cancer patients. We know so many people, each of us individually, people who are suffering from this disease. We already have been able to show that basic research done in a scientific laboratory can really provide a lot of information and help us develop new drugs for the treatment of cancer.
Yeah, I'm fine. Fine, I'm just fine, but a little emotional. I have to be very honest. This is such a special, special occasion for me. And I'm so delighted to be here today and really deeply honored to be one of the recipients of the 2019 Gairdner Awards, particularly on their 60th anniversary. And I think it's truly to Canada's credit that for the past 60 years, it has demonstrated its appreciation of excellence in science and recognized individuals throughout the world who have made major advances in biomedicine and in global health. So, as you've already heard, I've had a long-standing interest in natural products as sources for new medicines. And many of you may not realize that even today, in the United States and in Canada, 50% of the drugs that we use are of natural product origin. And that's amazing to me. So I've been interested in this area, and I've worked with drugs that came from bacteria, from plants, from trees, and even now we're working with an important new drug that comes from a sponge that grows maintained in the ocean. One of the things that Dr. Kokba said, and I thank you very much for your lovely introduction, was that I said that only a tree could make a drug of the structure of Taxol. That isn't completely true, although I said that all the time also. Today we know that life is much more complex, even in the production of a drug. And it is not the tree alone, but actually endophytes that live in this tree that together with the tree produce this drug. So we know that in this specific instance, there are fungi growing in the bark of Taxus brevifolia, the tree from which Taxol was isolated, and together they work to produce this drug. If you take the fungi out of the tree, it doesn't make enough Taxol. And the tree without the fungi can't make Taxol. And I think this is a very good example for scientists. We can't work alone. We are all working in collaborations, which are so important, not only within our own laboratories, with our students and our fellows, but across the lines of countries and around the world. Cancer is an extraordinary complex disease, and in order to make advances, we all have to work together and learn from each other and teach each other. And that is what I have learned, and that is the way that I try to work in my laboratory with my students and postdoctoral fellows. So again, it is such an honor for me to be here in front of you people and to receive this award, and to tell you that I really believe that working together and being advocates, not only of the work that we do in the laboratory, but being advocates of science, which is a part of each of our daily lives. And most people really don't know enough and appreciate science, and you are leading the way. Thank you so very much for this honor.
Thank you, Dr. Horwitz and Dr. Eves. I think both very inspiring words from our two laureates who are with us today. I'm so very glad that they're both here with us. And I will just say that we are heading up to Ottawa this afternoon, and tomorrow we'll be meeting parliamentarians on the Hill, actually at the Chateau Laurier, to have lunch and introduce them and have them tell their stories, and again, emphasizing how important science is, investment in science, from basic through to application, and we hope to make our case well in Ottawa. So now I'm going to continue. I want to continue now with the next Canada Gairdner International Awards, and the next one is Dr. Ronald Vail. And the citation for Dr. Vail reads, "For the landmark discovery of the motor protein kinesin and pioneering the understanding of its mechanism of action in driving cellular movement processes, including cell division and intracellular transport." Dr. Vail is professor of cellular molecular pharmacology. We're on a pharmacology kick this morning at the University of California, San Francisco. He's an investigator of the Howard Hughes Medical Institute, and you may know that he's recently been appointed executive director of HHMI's Genelia Research Campus and HHMI vice president, so he takes up that role in early 2020. Dr. Vail's discovery of kinesin and molecular motors have truly transformed the field of cell biology, telling us how living cells in our body generate motion. His work led to new tools for studying protein machines, contributed to the realization that motility defects underlie serious diseases of the nervous system, the heart, and other organ systems. So congratulations to Dr. Vail, who will tell us more about his fascinating work through his video.
The challenge that I was trying to solve is how living organisms generate motion. So motility is a fundamental attribute of life, and I wanted to know how it worked. In particular, I was interested in studying transport inside of nerve cells. So nerve cells are interesting because they're extraordinarily long cells. For example, a nerve cell could start in your spinal cord, and that one cell would extend a long process all the way down to your finger, and material has to be shipped from one end of the cell to the other, which is a motility mechanism very much similar to the way we deliver goods inside of a city. So in the quest to understand biological motility, we came across an entirely new machine that generates motion. This machine we ended up naming kinesin. It's a machine that's about 100 millionth of an inch in size, so extraordinarily small, but it works more efficiently than your car engine. And it ships goods inside of virtually all cells in the human body. So that was the initial discovery, and then we spent about 15 years trying to understand how this machine works, how it can take chemical energy and convert that into motion. There's so much we have yet to know about biological motility, and I hope the impact of this research and work by other people is that we can really understand this fundamental attribute of living organisms in even greater detail than we know now. And of course, there's also the possibility that we can help patients. In many cases, patients have certain diseases where there are defects in molecular motors, either in the heart or in other organs. And if we can understand how to manipulate these motors, we can perhaps treat their diseases. I think I would tell any young student who's considering science as a career that this is an awesome career. I think it's really important, first of all, to do something in life as a job that you really enjoy, that also has meaning, and science is one of those careers. I love my job, I love the profession, and I couldn't think of a more gratifying way to spend my life.
So, our next awardee is Dr. Timothy Springer. The citation for Dr. Springer reads, "For the discovery of the first immune system adhesion molecules, elucidation of their roles in antigen recognition and leukocyte homing, and translation of these discoveries into therapeutics for autoimmune diseases." So Dr. Springer is the Latham Family Professor of Biological Chemistry and Molecular Pharmacology. I didn't check this out. Here we are, third pharmacologist, and Professor of Medicine at Harvard Medical School and Boston Children's. His incredible work has changed our understanding of cell-to-cell interactions that control immune responses and the movement of leukocyte subsets out of the vasculature and into tissues. And that fundamental work, again, has opened the door, and he himself has been involved in this, opened the door for the first therapeutic use of antibodies to these cell-cell recognition receptors to treat autoimmune diseases. So his discoveries and research have really been transformative in the fields of cell biology and immunology. So congratulations to Dr. Springer, and we'll hear from him in his video.
The body of work I'm being recognized for is the discovery of adhesion molecules and homing molecules on lymphocytes. These molecules allow white blood cells to do their job to recognize foreign antigen, to mount immune responses to pathogens like bacteria and viruses. And they also allow white blood cells to home to certain organs in the body and areas where bacteria and viruses are causing a problem. And you might call these area code molecules. And I discovered that a series of 3 molecules, much like the 3 digits in an area code, in combination, are required to allow the specificity for the right kind of white blood cell to immigrate just at the right kind of site, be it in your big toe where there's a bacterial infection, or in your lungs where there's a viral infection. In those situations, we need different kinds of white blood cells. And I discovered the principles and the signaling molecules that allow the right kinds of white blood cells to get to the site where they can do their job. I'm very pleased about the direct impacts of my work on improving lives of patients. The molecules that I discovered that recognize other cells and the antibodies to them that I discovered have been made into drugs to treat many types of autoimmune diseases. I think my legacy will live on in several ways. One is the use of antibodies to explore functionally important biological pathways and use them to develop therapeutics. And I actually am helping in a personal way to get that going by founding a new institute, and we're developing not only open source reagents to help researchers discover biology and advance basic research, but we're developing therapeutics that we hope will be developed by companies to treat patients. Follow your dreams. I think the only way you can be successful in research is if you love it.
A common theme of love of science is certainly permeating all of the videos, and I hope permeating the room this morning. So our last two Canada International Awards go to, it's a shared citation, individual awards, to Dr. Bruce Stillman and Dr. John Diffley. The citation reads, awarded for their pioneering research on the eukaryotic DNA replication cycle, including initiation, regulation, and responses to DNA damage. So Dr. Stillman is the president of the Cold Spring Harbor Laboratory in New York, and Dr. Diffley is associate research director at the Francis Crick Institute in London. And together, their work really helps describe the exact sequence of events that are involved in how the DNA within our cells gets replicated during the cell cycle, once and only once. How is it replicated? How it's coordinated with other essential cellular events? And of course, understanding the basic features of DNA replication has great implications for understanding genomic instability and tumor heterogeneity in cancer. Dr. Stillman is most widely known for his fundamental work on the discovery of what's called the ORC protein complex that initiates DNA replication. Dr. Diffley, who started his career as a postdoc with Dr. Stillman, has built in his own career on this pioneering work and has most excitingly, I think, been able to actually reconstitute the entire DNA replication system from a eukaryotic cell in vitro, in a test tube, a true tour de force. So congratulations to Dr. Stillman and Diffley, and we'll hear from them in their video.
In 1992, my laboratory discovered the structure of the yeast origins of DNA replication. We showed that it was an unusual structure and that transcription factors could activate the initiation of DNA replication. At the same time, we discovered one of the key proteins that starts the entire process of chromosome duplication, a protein we call the origin recognition complex. The discovery of this complex really set the stage for the discovery of many, many other proteins involved in the initiation of DNA replication and how that process is controlled. So in every cell of our body, we have about 2 meters of DNA divided into the 46 chromosomes. And the rate of replication of DNA by the machinery is such that unless it started at many different places, it would take months to duplicate the entire genome. But in fact, it only takes about 8 hours. So you can imagine an enzymatic machinery, which we've helped discover, copying molecules of DNA that are that long every minute of our lifetime.
I think the main thing we're being recognized for is really working out in molecular detail how our genomes are copied exactly once in every cell cycle. Our genomes use a divide and conquer approach to their replication. So replication actually begins from many, many sites along the genome. In a typical human cell, that's probably around 50,000 or 100,000 of these replication origins that get activated in each cell cycle. And the challenge really was to try and understand how all of those replication origins can be used exactly once in every cell cycle, but never used more than once. Because of course, if they were to reinitiate replication, you get a partial reduplication of your genome. So that was really the challenge. And so we've invested a lot of time over the years in reconstituting this process of replication. We first built the helicase onto DNA, and then we've been able to activate the helicase and build the entire replisome. I think that's had an important impact on the field in terms of providing a tool for understanding how many of the processes that are coupled to DNA replication work.
So I think the impact of the work that we've done is to help identify the entire machinery that replicates the genomes of cells. This is really fundamental knowledge, but it can potentially impact the understanding of new treatments of cancer. Many, in fact, current chemotherapies target enzymes or the process of DNA replication. And these are fairly blunt instruments for cancer, and of course the future is to identify much more surgically precise treatments that are not as toxic.
So congratulations again to Dr. Stillman and Diffley. And now I'm going to announce the 2019 John Dirks Canada Gairdner Global Health Award. And this award is named, as you can tell, in honor of our President Emeritus, Dr. John Dirks, who's with us here today at the front. So it's always great to see you here, John, and congratulations again. The 2019 John Dirks Canada Gairdner Global Health Award for outstanding achievements in global health research this year goes to Dr. Vikram Patel. And the citation reads, for his world-leading research in global mental health, generating knowledge on the burden and determinants of mental health problems in low and middle-income countries, and pioneering approaches for the prevention and treatment of mental health in low-resource settings. Dr. Patel is currently the Pershing Square Professor of Global Health at Harvard Medical School. His work and research is really focused on raising awareness of mental health as a global problem. His novel approaches to community-based interventions in mental health have had significant impact in raising awareness of mental health as a major global challenge. Those of you who remember our awardees last year, Dr. Lopez and Murray, on the global burden of disease, may remember that in fact mental health is one of the major individual determinants of health and well-being worldwide. So his work has raised the impact and raised awareness of mental health. He's had impact on research, teaching, policy, and practice agendas. For example, the Grand Challenges in Global Mental Health Research Initiative, the World Health Organization's flagship program in mental health, and India's first national mental health policy. But I think importantly, the approaches that he's pioneered in low and middle income countries, low resource settings, are actually increasingly relevant to our homegrown issues around access to mental health support in vulnerable populations in Canada. So congratulations to Dr. Patel, and we'll hear from him in his video.
The challenge that I've tried to solve is the fact that the vast majority of the world's people are affected by mental health problems, most of whom live in low and middle income countries, do not receive the care that we know can transform their lives. This is because of many reasons. For example, the idea that mental health problems are too trivial for poor people in poor countries. They have far more important things to worry about. Or that to address mental health problems, we need specialists who are very scarce and very expensive, and most countries and most people simply cannot afford them. My work has tried to address these ideas by generating knowledge that demonstrates, for example, that mental health problems are universal forms of human suffering, that they're profoundly associated with social disadvantage and deprivation, and indeed when one is affected by a mental health problem, it becomes doubly difficult to address that disadvantage. But perhaps most excitingly, the demonstration that resources that are plentiful and affordable in every community can be harnessed to address mental health problems. For example, through the use of community health workers to deliver psychological and social interventions. I believe that my work, alongside that of others, has made a contribution to the emergence of global mental health as one of the most dynamic and vibrant fields of global health. It has also influenced national and global policy to prioritize mental health as a legitimate cause of human suffering for investment and action. And I'm also very proud that I have played a role in helping bridge the gaps between people with a lived experience and professionals to work together with mutual respect to address this challenge. Looking ahead, I would hope that my work will contribute to alleviating the global burden of suffering due to mental health problems, both by improving access to quality care, as well as by preventing mental health problems. When I received the call telling me that I was this year's recipient of this incredible prize, I was speechless, because I knew I was following in the footsteps of giants of global health, many of whom I consider my heroes. I was truly humbled.
So there we have it, 7 outstanding individuals who now join the Gairdner family, a distinguished group of researchers responsible for amazing, significant advances in science. And you heard their passion, you heard their excitement, you heard their desire that the next generation of researchers remembers and is engaged in that excitement as a great career. And a large part of our core mandate really is inspiring the next generation of researchers, innovators, and leaders through our Pan-Canadian outreach program. And over the course of this year, Gairdner will send our laureates across the country to share their science and their stories with faculty members, trainees, high school students at our partner universities, and at symposia and public lectures. If you watch our website, you'll get a full listing of all our events. Special 60th anniversary content will be coming up soon on the website, and you can subscribe to our newsletter to get more updated information. I also want to encourage those of you who are in the room who are engaged in science yourself, recognize the importance of the recognition of excellence that Gairdner provides, and I want you to encourage you to consider future nominations of outstanding scientists from Canada and around the world for a Gairdner Award. We are particularly interested in increasing the diversity of our nomination pool, so please think broadly and bring forward great nominations for future winners. The nomination portal for 2020 awards opens tomorrow on our website. So get ready. And before you head out, if you are not already on our list of contacts, you can pick up the Gairdner pen that you have on your table and fill out the sign-up card at your seat, and we'll put you on our list. You'll stay connected, and you can join us again at the announcement again next year and our events throughout the year. And I encourage you all to think about buying a ticket for the gala, which is, of course, the great event that takes place in October, when we truly celebrate all the awardees, hear their stories, and have a really great party at the Royal Ontario Museum. So thanks for coming this morning, and we hope to see you again soon.